The project we're going to work on today was developed by the Allen Institute for Brain Science, which was founded in 2003 by one of the Microsoft co-founders, Paul Allen, and now everybody calls this project the Allen Brain. This is a privately funded organization that focuses on researching the brain, but they also produce some cutting-edge online research tools that are open to all. Do look around when you have a chance. They often put out newsletters of the research that they've done recently, but we will move on directly to the brain map page. So this is what we're going to do today. We're going to build a 3D brain that is going to look similar to what you see here. You can reach this page by directly typing brainmap.org or by searching for it in a search engine. Here you can see all of the ongoing projects from the Allen Brain Institute. Remember that these are all open to everyone, but we're going to start with the reference atlases. So please click on the reference atlas. First, I'm going to show you an adult human gyral atlas. Let's click on this. A reference atlas is a detailed map of a brain, and we used a paper version of this back in my day, and the paper version of the human atlas is still used today. What you're looking at here is a slice of a human brain with all the different parts labeled. By just hovering over a particular part of the brain, you can learn their names. So here's the superior temporal gyrus. Here is a middle temporal gyrus, the putamen, the caudal part of the cingulate gyrus, and so on and so forth. You can zoom in and out to see the entire brain a little bit better. And you can move from the front of the brain to the back of the brain, if you wish. Now, this is a very useful resource, so do look around when you have a chance. But this is a two-dimensional map of a very complex three-dimensional structure. So what we're going to focus on today is a 3D map of the brain. Now let's go back, and this time click on the Adult Mouse 3D Viewer. You can also reach this page by going to connectivity.brain-map.org or just by searching for the Brain Explorer. Here we will build a 3D mouse brain. Essentially, this is a 3D version of a reference atlas. I will first show you how to navigate the interface. Let's focus on the panels on the left side of the screen first. So here you have three major groups of structures that we can build. The fiber tracts, the white matter. This is how the brain is connected. So let's click on this. Let's click on the little eye icon. And all of the white matter tracks are now loaded. We can also load the ventricular systems. These are the pools of the cerebrospinal fluids that are located in the brain. And finally, we can load all of the gray matter structures, essentially the cells. You can use your mouse to zoom in and out. You can turn the brain in every direction as you like. We can get rid of this reference grid, and we can toggle transparency so we can see internal structures better. Of course, with this, you can't really see much detail because we just loaded everything that we have in the brain. So let's explore the brain in a little bit more detail. I'm going to hide all these structures again. And we're going to focus just on the gray matter. Do you see this little triangle next to this third set of structures? This is a toggle down button. So if you click it, you will see all of the structures that are associated with this major division of the brain. If you hover your pointer over the name, you will see both the abbreviation and the full name. An abbreviation is something you might see in a textbook. Every time you see a little triangle pointing downward, or pointing to the right rather, that means there are more structures that are associated with this particular major division. So you can click on them to see finer and finer detail of the brain. So let's build a few parts. Here we are going to build the dorsal auditory area, layer 1. Layer 5, 6A, and 6B. I'm going to toggle the transparency off so we can see a little bit better. We can build parts of the isocortex. We can load the entire substructure. Or build it layer by layer. Let's build the entire olfactory system in a mouse. And you can see it's very large. That's because mice rely on their sense of smell much more than humans, for example, do. So therefore, the part of the brain that processes the olfactory information in the mouse is fairly large. Let's also build the hippocampus. The hippocampus is a very interesting structure. This is the part of the brain that is important for learning and memory. It's a very complex structure, and it has been extensively studied in both mice and humans because this is one of the parts of the brain that deteriorates in Alzheimer's disease. You're learning something new now, so your hippocampus is probably very active right now. Notice that the mouse hippocampus is comparatively large. So you can construct the mouse brain piece by piece, or we can toggle everything off. And instead deconstruct a particular structure. If you go into the structure sets, you can load a hippocampal formation, for example, that we just looked at. And now by toggling the visibility of these structures, you can figure out exactly where they're located in relation to the other substructures in the hippocampus. So for example, I'm turning off the dentate gyrus, and that is a fairly small structure that's nestled in here. Now, this resource gives you an ability to build structures and learn their names, but of course, you will still have to go on the internet to find out what they do. Go on the internet or a book. So now that you know how to operate the Allen Brain Explorer, let's do an actual experiment. We're going to close this page, go back to the list of projects, and this time click on the Mouse Connectivity Atlas. So here you see a mouse brain with many colored dots. Before we go any further, I want to pause and acknowledge that what you see here was done by experimenting with animals. I want you to know that we consider the reasons for using animals in experiments very seriously, and only where no other alternative exists. The experiments have to be done humanely. This is governed by the law, and the existence of these resources reduces the need for other scientists to do these experiments because the data is already here. This is something you should consider if you want to pursue science further. You will encounter description of animal experiments, although as a field, we are moving steadily away from this. In this experiment, we're going to figure out which parts of the brain are connected to each other. The brain is very interconnected. This is the main function of the brain. It is to store, process, and relay information, a lot like a computer. Let's say you're dancing. You need to remember dance moves you learned before, perhaps create new ones. You need to coordinate your movement with the music. You also need to see so you don't bump into someone else. This means during dancing, there's information exchange between the parts of your brain that coordinate movement, balance, and process what you see and hear, and probably many more. You have seen how complex the brain is, but it is also very important to understand how it is connected. There are very few ways we can do this. So let me explain what you're looking at. Each colored dot you see here represents an injection that was made into a mouse brain. The colors match those you've seen in the 3D reference atlas. You can turn the brain around by using these scroll buttons. The mouse was injected with a genetically modified virus. This virus can still infect cells, specifically it infects neurons, but it can no longer make copies of itself and infect other cells. We've taken the DNA out. Instead, the virus now carries a DNA from a bioluminescent jellyfish. In other words, the virus now carries instructions to make a cell glow. It carries a protein you probably heard of called the green fluorescent protein, or GFP. When we inject the mouse with this modified virus, it will be able to enter a few neurons near the injection site, but it will not spread further. The neurons the virus enters will now glow green. The injection is made into the gray matter where the cell body is located, but the entire neuron will glow, and we can trace where the axon goes, the part of the neuron that connects to the other parts of the brain. So pick a new dot, and if you hover over it, it will give the name of the structure where the injection was made. Bigger circles mean that part of the brain is very connected. Smaller circles, it's not as connected. So I've picked one in the motor area. This is the part of your brain that controls your movement. And what you can see here is the image of the injection site and the reconstruction of the connectivity of this particular area. You can see where the axons go. Let's click on a small 3D button in the top right corner. Select the web viewer. And you will be taken to the now familiar Allen Brain Explorer page. It may take a little bit of time to load it, so please be patient. So here So here you can see the injection site in these big lumps. These are the cells that were infected with the virus. And all of the white lines are the axons that these cells sent to the other parts of the brain. You now notice that you have an experiment loaded into the right panel of the screen. This program has no save function, but you will be able to reach your experiment again by just typing this number into the search panel here. So your job will be, and this will take some time, so please do this on your own, is to figure out where does your injection projects to. So by clicking on each of these little squares, if you click on a square, you will see the area this particular neuron projects to in the experiment information. So this particular axon terminates in the primary motor area layer 5. So in order to build this, you need to either find it in the anatomical ontology page section, or you can simply put in the abbreviation into the search bar. Click on it and make it visible. And here it is. And you can repeat this several times to build your brain. Remember to toggle the transparency on so you can see the structures below. So now I'm just going to give you information and quick instructions you can refer to. So here are the websites if you want to reach the resources directly. Please take a screenshot or write them down if you like. Here is a quick reminder of how to operate the Allen Brain Connectivity Atlas. Just follow steps 1 through 4. There are other functions you can explore here, and please feel free to do so. But some of them are much more complex than others and require quite a specific set of instructions. And then finally, here is a quick reminder of how to navigate the Allen Brain Explorer interface. Thanks so much for listening. Bye-bye.